This is Chapter 14, Neurobiology of Sleep, and it corresponds to Chapter 13 of your textbook, including Section 7. No announcements or reminders at this time. As I mentioned in the previous lecture, I'm recording a lot of these lectures ahead of time, so I will keep everyone updated about announcements through the Laulima announcement portal. And your not required reading is an article from Nature on a giant neuron that was found wrapped around the entire mouse brain. So in that image to the right, this is a mouse brain, and there was one neuron that took up a lot of space and covered a lot of areas. And the authors think that it might be a piece of the puzzle in understanding the neural underpinnings of consciousness, which would necessarily need to be a way of connecting many different brain areas so that they can work together.
Moving on to Chapter 13, the Neurobiology of Sleep, we're going to cover sections 1 through 6. Okay, Section 1, Biological Clocks. Biorhythms are inherent timing mechanisms that control or initiate biological processes. These are typically linked to external cycles, such as the night-day cycle or across the seasons of the year. If you remember back to one of the early chapters, humans adapted in Africa, in the East Africa region, which is right by the equator. So we adapted in a more balanced night-day cycle, but we have adapted to living in areas where the night-day cycle is not as balanced. So in areas very far north, such as Iceland, the nights can be very long in the winter, the days can be very long in the summer, and humans have adapted to this. Although there are some drawbacks and issues that people experience. Even very simple organisms show biorhythms, indicating that this is a function of organisms that showed up very early in the evolutionary process. And biorhythms affect a lot of things, including blood pressure, body temperature, metabolism, sex drive, and hunger. Humans are considered diurnal, which means that we're awake during the day, and this is in contrast to nocturnal, which means awake during the night, such as raccoons or some cats. Biological clocks are guided by internal and external cues, and they prepare the body for upcoming expected events. So they prepare the body for sleep, drops in environmental temperatures that happen at night, for example, or with the seasons. There are different types of biological clocks. Ultraradian rhythm is a biological rhythm that lasts less than 1 day. So examples of this is like eating or activity. So we tend to have like rest, active cycles that cycle a few times throughout the day. Eating tends to happen 3 to 4 times in a day, or maybe 2 to 4 times in a day. Circadian rhythm is the day-night cycle, and this sleep is the prime example of this. The sleep-wake cycle happens once throughout the day and night for 24 hours. Circannual rhythm are biological rhythms that cycle over the year, over the 4 seasons, and this we see mostly in animals, so migration, yearly migrations, mating seasons, hibernation. And the circulunar cycle are biological rhythms that last 1 month, so 1 lunar cycle. An example of this would be in human women, the menstrual cycle happens about every 28 days. Human circadian rhythms actually lengthen in the absence of external cues, so a typical circadian rhythm would be 24 hours. With the absence of light or external daylight, this can lengthen to 25 to 27 hours. There's a German term called Zeitgeber, which means time giver, and this is any kind of external cue that helps to set these biological clocks. So a typical one would be light, another one might be temperature. The invention and popularity of artificial light likely has contributed to increased sleep issues in humans. It allows us to stay up and stay active after the sun has gone down. This probably has contributed to metabolic issues such as obesity and diabetes, as well as mood problems associated with difficulty sleeping and not enough sleep. People who work night shifts also have issues, higher levels of metabolic issues, again, such as obesity, diabetes, and this is likely because of decreased quality and time of sleep. Also, long-distance air travel can cause kind of insidious issues in the circadian rhythms. People who have bipolar disorder, which is actually strongly affected by sleep. So if the circadian rhythm is disrupted in people who have bipolar disorder, such as if there's a time zone change, this can actually trigger mood episodes, because it can trigger more typically mania, but also depression in people with bipolar disorder. And then long-distance relationships can actually cause circadian issues. So if somebody has a long-distance relationship that where their partner is, say, up to like 6 hours different, there's actually some evidence that the person sort of emotionally is in that other time zone, and so this can cause issues with their circadian rhythm. Okay, and this is just some examples of disorders that can happen when biological clocks, particularly circadian rhythms, are not functioning normal. So there's delayed sleep phase syndrome, which is when somebody has a significantly later bedtime than what's normal, later than a social norm. And advanced sleep phase syndrome, where somebody goes to bed extremely early. And many of these things happen in clinical populations. I won't read through them all, but you're welcome to if you're interested. Okay, moving on to section 2, the neural basis of the biological clock. So in this section, there's a lot of detail that we could cover. I decided, since we're doing 2 chapters at once, I'm going to go over some of the topics with more broad strokes, specifically the suprachiasmatic nucleus. So don't worry about all the details in the book unless that's something that you're personally motivated to learn about. The suprachiasmatic nucleus, or the SCN, is the main regulator of circadian rhythms. And this is a nucleus, or a group of neurons within the hypothalamus. And if damaged, the sleep-wake cycle is very severely disrupted. That's how we know that the SCN is responsible for the sleep-wake cycle. Interestingly, the feeding schedule is not significantly affected, so the shorter cycle is not as affected, meaning that the SCN is probably very specific to this circadian rhythm, the sleep-wake cycle. SCN cells have actually like an intrinsic rhythm of activity, so just by the way that they're built, they are set like a clock, and the timing of this clock is mostly genetically determined, but there are external factors. So things like external light, things like that contribute to the entrainment of the SCN cells. Information about light comes from the visual system. The optic chiasm, which, if you remember from the visual system, is where the nerves from the eyes, the optic nerves, cross over as they move back toward the visual cortex. While they're moving back, they also have some projections to the SCN, which gives the SCN information about whether there's light in the environment. The SCN cells are also influenced by physical activity and feeding cycles, and all of these things sort of entrain the SCN cells, so it's like setting a clock or setting your watch. Different types of people have different types of chronotypes. Chronotypes are varieties of circadian rhythms. So you might have heard like an early bird or an early lark is active in the morning, night owl is active in the evening, and there are actually both genetic and behavioral influences that determine chronotypes. So some people just cannot stay up at night. They have to go to bed by 9 or 10 p.m. Other people it's really hard for them to go to bed before 12 or 1 a.m. And while this might be influenced by some behaviors, some of this really is genetic. People can also differ in the length of their circadian rhythm. There's a pretty debilitating disorder where somebody has a significantly longer circadian rhythm than 24 hours, which means that they're going to bed a little bit later every single night. And that can be pretty stressful and pretty difficult to fight that urge if they're sort of set at a longer circadian rhythm. So the SCN functions as a pacemaker. which in turn entrains or directs what's called slave oscillators, and those slave oscillators control the functions that are regulated by their circadian rhythm. So slave oscillators control motor activity, different ones control eating rhythms, and then other slave oscillators control body temperature. The SEN projects axons to other nuclei in the hypothalamus and the thalamus, which connect to other areas of the brain and body. So SEN actually has a lot of projections and influences many different functions within the brain and body. SEN also connects to the pituitary gland, which regulates hormonal activity and also directly releases some hormones by itself, although most of the hormonal activity is through the mediation of the pituitary gland. And then the SEN also projects to the pineal gland, which inhibits melatonin during times of activity. So I'm going to cover melatonin in a little more detail later, but you might be familiar with melatonin because it can be taken as a supplement for people who want to entrain their circadian rhythm. People who maybe were on the night shift and need to switch to the day shift might take melatonin to kind of induce sleepiness. Circadian rhythms are also regulated by the SCN. So in animals, the SCN regulates mating seasons by controlling hormone release in the pineal glands. Shorter days means less daylight. Less daylight means less inhibition of melatonin, so it means more melatonin in the system. Melatonin inhibits sex hormones, and this leads to fewer reproductive activities. So it's very interesting, this connection between less daylight and fewer reproductive activities. The result of this is that animals who have mating seasons, like in the spring or the summer, for instance, are not reproductively active during the winter, during the fall, so they're not having babies that will be born in the winter, which would obviously, at least in areas where the winter is cold, this would be more dangerous for babies to be born during this time. Cognition and emotion also vary rhythmically. There's not a ton of research on this, but there is some data that, especially within the circadian rhythm, within the sleep-wake cycle, cognition and emotion vary. So, for instance, if you look at the top graph, there's some in the pre and post-adolescence. Adolescents tend to be different in general in terms of their circadian rhythms. If you know, like high schoolers and middle schoolers, they kind of tend to stay up later as opposed to the rest of the population. But for pre and post-adolescence, there's this morning tends to be a time of peak alertness with a dip right after lunchtime, and then it comes back up in kind of maybe the mid to late afternoon, and then steadily declines toward what is a typical bedtime. Emotions can also tend to occur at the same time each day. One example is this time in the mid-afternoon, sort of after lunch, when there's also this cognitive dip. There can be an emotional dip. People can feel a little bit down or grumpy during this time. There is an interesting study where people took data from Twitter, from UK Twitter accounts, and looked at, based on words that were associated with joy or sadness, looked at what time of day people were more likely to tweet things about, like happy tweets versus sad tweets. And so they found kind of, first, just this peak in general of tweeting. It looks like just a little bit before lunch, maybe at the 9 to 10 a.m. time. And then also fewer as the day gets longer, like more toward 9 p.m., fewer joy tweets as opposed to sadness tweets. So the comparison of these two, tweets could indicate, one interpretation might be that people in general are feeling a little bit sadder and a little less joy as the day goes on. Some psychiatric disorders might also include a bidirectional relationship with the circadian rhythm. So people with bipolar disorder, depression, schizophrenia, if they have an increase in the symptoms, mood symptoms or psychotic symptoms, it's likely to mess with their circadian rhythms. So for example, people with bipolar disorder, if they have a manic episode, If it's a serious manic episode, they're sleeping very little, their circadian rhythm is getting completely messed up, and then they tend to crash, go into a depressed episode, and might be sleeping for much longer, or paradoxically, other people with depression are not, also have difficulty sleeping during the night, or at least getting good quality of sleep. People with schizophrenia as well tend to do what we call like a day-night shift switch. So a lot of times when people are experiencing active psychotic symptoms, they're active during the night and sleeping. They're sedated during the day. So section 3, this section's going to differ a bit from the book. I prefer a slightly different, and I think maybe a little bit updated way to present the stages of sleep. So measuring sleep in general, individual differences are normal. Sleep differences are not a problem typically unless it's causing distress for a person or causing physiological issues. Some people only need a couple, like 5 to 6 hours of sleep. Other people need 9 to 10. Two main ways of measuring sleep, if somebody's having sleep issues, is just keeping a diary of sleep, as well as things that may influence sleep, like caffeine intake, napping, foods, things like this. And then EEG, a sleep study. And EEG is sometimes accompanied by measurement of the facial muscles and the eye movement, but typically just EEG is enough in a sleep study. Okay, so stages of sleep. To compare, awake, there's two general types of awakeness. There's being fully alert, and in this case the EEG is small amplitude, so the waveforms are not super high, not very tall, and it's high frequency, meaning the waveforms are close together. So this is 15 to 30 hertz, and these are called beta rhythms. In this frequency range, these are called beta rhythms. When somebody is drowsy, and typically when their eyes are closed, when they're feeling relaxed, the waveform becomes more rhythmic and also a lower frequency, so more space between the peaks and the troughs of the EEG signal, and these are called alpha rhythms. They oscillate at 7 to 11 hertz typically. In terms of the stages of sleep, sleep typically occurs in cycles of about 90 minutes. And within those 90 minutes, there's two main types of sleep. There's rapid eye movement sleep, or REM sleep, and there's non-rapid eye movement sleep, or NREM sleep, or non-REM sleep. There's 3 to 4 stages of non-REM sleep. There used to be, I think kind of the old system is more that this conceptualization of 4 stages. There's a newer system where stages 3 and 4 are kind of pushed together, so either one is correct. For the sake of being consistent with the book, I'll talk about stage 3 as if it's stage 3 and 4, and let me know if you find that confusing. Okay, so in non-REM sleep, in which there are those 3 to 4 stages, stage 1 is the beginning of sleep. So there's slow eye movement, but the eyes are closed. These alpha waves disappear and are replaced by theta waves, which are a little bit slower, 6 to 10 hertz, and look something like this picture up here. People often twitch, their muscles twitch, and if the twitching is severe enough, they wake up, and a lot of times if you wake up somebody in stage 1 sleep, they believe they haven't been sleeping, so they're not even aware they've been sleeping. During this stage, during several stages, something called hypnagogic hallucinations can occur. Most likely during stage 1, hypnagogic hallucinations, as long as they're minor, are actually rather common, and this is hearing or seeing something that doesn't exist. So hallucination is perceiving something that doesn't exist. It's sort of like a mini dream. A common one is people will suddenly feel like they're falling and wake up. Another one is hearing their voice being called. That's a pretty common hypnagogic hallucination. And just to reiterate, these are very common. This is not at all a sign of psychosis. It's just a normal part of sleep. Stage 2 is slightly deeper sleep. Eye movements have stopped. sleep spindles occur. Sleep spindles are these high frequency bursts, these little bunches of activity. And then in stages 3 and 4, which is, as I said before, has kind of been collapsed into stage 3 in more modern conceptualizations of sleep. This is deep sleep. This is what you hear like slow wave sleep. What's called delta waves occur, very slow, 0.5 to 4 hertz. And sleepwalking and a lot of other parasomnias or sleeping disorders that occur during sleep are most likely to occur during this phase. And then there's REM sleep. This is also called paradoxical sleep because the EEG in REM sleep looks surprisingly similar to wakeful EEG. If you look on this purple graph to the right, at the very top is wakeful EEG with this high frequency, low amplitude. And similarly in REM sleep at the bottom, you see this high frequency, low amplitude. There are distinctions. REM sleep tends to be more rhythmic than awake sleep, but they look surprisingly similar on EEG. So REM sleep is, of course, rapid eye movement sleep. It's characterized by rapid movement of the eyes. You can see the eyes moving underneath the eyelids. And vivid dreaming is more likely to occur during REM sleep. Dreaming can occur during any sleep cycle. It's a little more common during stages 3 and 4 and REM sleep, and you're more likely to remember it during REM sleep. Other indicators, physiological indicators besides EEG that somebody is sleeping are decrease in metabolic activity, decrease in body temperatures, and then breathing and heart rate changes, even between the sleep cycles, tends to slow down as people get progressively deeper into sleep, although in REM it can speed up again. As people get older, the amount of sleep they need typically decreases, and the proportion of sleep that is dedicated to REM actually decreases as well. Many animals are able to maintain posture so they can remain standing, for example, during non-REM sleep, but muscle tone is completely relaxed during REM sleep in all animals. And you can see up in the right, upper right-hand corner, these are two PET images of a brain during non-REM sleep and REM sleep. You can see that the brain is more active during REM sleep, kind of consistent with what we see on the EEG. So dreaming is, of course, one of the most interesting aspects of sleep. We don't know much about dreams. Actually, we don't know that much about sleep in general. A lot of what we know is theories that can be difficult to substantiate. Dreaming, as I mentioned before, can occur in both non-REM and REM sleep. Dreams in REM are more vivid, more memorable, and we dream more often in REM as compared to non-REM. The longer we sleep, the more we do dream, so the further we get through the night, the more likely we are to dream. Almost everybody dreams, although many people don't believe that they dream. It's more likely that they just don't remember their dreams, so they wake up at a time that they're not remembering their dreams. And contrary to what a lot of people are, there's kind of a common myth that even if a dream seems to take course over 20 minutes, it actually happened instantaneously. It actually appears that dreams more often occur in real time. So if you dream an event happened over the course of 10 minutes, it might have actually been 10 minutes of dreaming. In terms of the function of dreams, again, a lot of this is rather theoretical. It's hard to substantiate and hard to test a lot of these things experimentally. The first person in modern psychology to really talk about dreams was Sigmund Freud, who's considered the father of psychoanalysis. And in Freud's view, dreams are representations of these unconscious wishes, and that we can interpret these dreams and get an understanding of people's unconscious wishes. As is the case with much of what Freud said, most scientists do not agree with this anymore. There's really no reason to believe this. Freud had poor methodology. He used his own patients as subjects, and Freud wasn't much of a, he was more of a philosopher than he was a scientist. So kind of in general, it's best to be very skeptical about Freud's theories. Another possible function of dreams is the activation synthesis hypothesis, which says that dreams are completely meaningless. They're just random firings that happen in the brainstem, then activate other brain areas that are involved in emotion memory, and so the brain, which is, our brain is always trying to make meaning of all our sensations, and so the brain kind of makes meaning out of these random firings. Another hypothesis is the coping hypothesis, which says that dreams help us prepare for life-threatening events. Evidence for this includes that the fact that dreams have mostly negative content, and this has been tested empirically. People are awoken while they're sleeping and asked about what their dream was, and most dreams have negative content. As well, scientists have noted that in dreams, when somebody is faced with a threatening situation, they're more likely to approach. So in the fight or flight reflex, they're more likely to fight as opposed to avoid that threatening behavior. And so the idea in the coping hypothesis is that we kind of practice dealing with threatening situations through our dreams, which is kind of a safe space. Lucid dreaming is an interesting phenomenon you might have heard of, and this is dreaming in which one is aware that they are dreaming and they can control the dream to some extent. So if someone is having a lucid dream, they might be able to decide that they're going to start flying in the dream and then fly. And they know that it's a dream, they know it's not real, but they can kind of imagine or control the situation, sort of like virtual reality. Lucid dreaming the difference between regular dreaming and lucid dreaming may be the activation of the dorsolateral prefrontal cortex, which is this area of the prefrontal cortex that's involved in working memory, as well as kind of other executive functionings. So there's some indication that that might be active during lucid dreaming, but not active during the rest of REM sleep or regular non-lucid dreaming. Lucid dreaming might also be associated with micro-awakenings, so people might be waking up quickly, then going back to sleep during lucid dreaming, and so kind of walking that threshold between awake and sleeping. And preliminary studies do indicate that teaching lucid dreaming can be used to reduce nightmares in people who have very severe and debilitating nightmares. I haven't tried this, and I haven't looked too much into the research, but it does appear that you can train yourself to lucid dream. And it's surprisingly a bit similar to some techniques that were used in that movie, what's it called, Inception. So if you're interested, you can look at this YouTube link, and there are other resources online, but they all have the same general strategy, which is sort of a reality testing strategy, that you teach yourself while you're awake, and then kind of get it in your brain enough that it might show up when you're dreaming. Okay, so moving on to section 4, what does sleep accomplish? We actually don't know for sure that much about why we sleep, evolutionarily why we sleep, but we have some ideas. So sleep appears to be a biological adaptation. We save energy during sleep, during the nighttime. We can't, as diurnal animals, we can't forage for food or move around easily during the nighttime, so it makes sense for us to sleep and save energy. Evidence for this is pretty clear data that indicates that predators sleep longer than prey. Predators can afford to sleep longer than prey because they don't have to be awake and alert, at least as much as prey. There's also basic rest activity cycle, the basic rest activity cycle, which is cycles of 90 minutes in humans. And these 90-minute cycles appear to be present in both sleep and wakefulness. So as we covered just a couple slides ago, the sleep cycles, the 3 to 4 non-REM cycles and the REM cycles tend to occur in 90-minute intervals, and our day cycles also happen to occur in 90-minute intervals. So we tend to have like activity rest cycles that occur in these 90 minutes. Sleep is also appears to be restorative for our body and our brain. And this evidence comes mostly from looking at what happens when people don't sleep. So acute, extreme sleep deprivation can lead to pretty serious cognitive problems, memory and attention. It can lead to mania, even in people who have not previously had mania. So sleep deprivation can actually lead to an inability to sleep and kind of hyperactivity. It can lead to hallucinations, seizures, heart problems, and proneness to accidents. When people are deprived specifically of REM sleep, when they're allowed to actually have REM sleep later, their REM sleep becomes longer, and this appears to indicate that the body needs REM sleep because it's kind of compensating for that deprivation of REM sleep. Antidepressants can suppress REM sleep, which might be one reason that people tend to be a little more groggy when they're on antidepressants. Just a quick note at the top about a typo in your book. We're going to cover memory in a later chapter anyway, and I'm not going to get too much into memory before then, but just if you were confused by that typo, there's a quick clarification. Alright, so the role of sleep in memory storage. There's been quite a bit of attention to the role that sleep plays in specifically the consolidation and recall of memory. So memory improves after sleep. The brain areas for functions that are learned before sleep are activated during sleep. So if people learn a motor task repetitively before they sleep, and then their brain activity is recorded while they sleep, those areas that were used during the learning of the motor activity are activated during sleep, indicating they might be sort of encoding or doing something with that memory during sleep. There are three main phases of memory. There's the labile-only phase, where memory is kind of like briefly encoded, but it's not permanent in the brain yet. And then there's consolidation, where memory is stored permanently, and recall, where memory is brought back into conscious awareness. And those latter two phases of memory are implicated in the sleep process. So theories of memory function in sleep, there's actually a lot of them, but some three common ones are the multiple processes, which is the idea that different kinds of memory are stored during different sleep phases. In the sequential processes theory, different stages of memory consolidation and recall occur during different sleep stages. In the storage process theory, waking memory functions continue in the same brain areas during sleep. The advantage is that memory functions don't have to compete with other waking functions, so it's kind of a time when memory can take over the brain and do a lot of its work while somebody was sleeping. Interestingly, marine mammals, including dolphins, whales, orcas, porpoises, have unit hemispherical slow-wave sleep. So that means during non-REM, during that 3 to 4 sleep phase, one hemisphere of the brain remains awake while the other one sleeps. So they sleep one hemisphere at a time. And the eye contralateral to the awake hemisphere remains open. So they literally sleep with one eye open. This is likely in part because they have to maintain some postural control because they are underwater. They can't just lose all muscle tone. And for this reason as well, they may not have REM sleep. REM sleep is associated with a complete loss of muscle control and may not be adaptive for marine mammals. Okay, so section 5, the neural basis of sleep. Again, I'm going to deviate from the book a little bit in this chapter. The induction and maintenance of sleep involves quite a variety of brain areas, different hormones and neurotransmitters. We're not going to cover it exhaustively. I'm just going to cover the main ones. So one of the main areas is the reticular activating system, or the RAS. And this is also known as the reticular formation. So our reticulum is a large group of cell nuclei and nerve cell fibers. The RAS runs through the center of the brainstem specifically, and it projects to the hypothalamus, which in turn projects to other areas of the brain. Sensory stimulation creates wakefulness by stimulating these RAS neurons in the brainstem. Brainstem damage can be very serious and often leads to coma, and this is in part due to the RAS. If somebody has damage to their RAS, they can't be awakened. They can't be stimulated into wakefulness. Many neurochemicals are involved in sleep, and we actually don't understand a lot of the complex processes that these are all involved in, but many of the monoamines, norepinephrine, dopamine, serotonin, are involved. But I'll just kind of mention some of the main actors. So cortisol, levels of cortisol rise before waking and kind of peak right after we wake up, and then decrease in the evening, slowly rising again, starting in early morning if you're someone who has a normal sleep schedule. Serotonin converts to melatonin in the pineal gland. Melatonin likely promotes sleepiness. The exact function is not totally clear. If you've ever heard of tryptophan, this is a compound that there are high levels of tryptophan in many like protein-based foods, such as I think like fish and egg whites. So tryptophan converts to serotonin, which converts to melatonin, which you might have heard that tryptophan induces sleepiness, and this is the process by which tryptophan induces sleepiness. There's a myth associated with Thanksgiving that turkey's really high in tryptophan, and that's why you get sleepy after the Thanksgiving meal. This is actually not true. Turkey's not particularly high in tryptophan, and unless you're eating like a huge plate of turkey, it's more likely that you're just eating a ton of carbohydrates, which convert to serotonin, and also just overeating in general makes people sleepy. So unfortunately, it's not the turkey. You can't blame the turkey. It's just overeating that's making people sleepy. Adenosine is a compound that we covered just briefly in the hormones and neurotransmitter section. It's a natural byproduct of energy use, or ATP, in our cells. And throughout the day, as we use energy in our cells as ATP is used, there's a buildup of adenosine that creates a pressure to sleep, or an inhibition of wakefulness. If you remember, caffeine is an antagonist of adenosine. So caffeine suppresses adenosine, which helps us stay awake. Section 6, sleep disorders. There's actually a ton of sleep disorders, but I'm just going to cover some of the more common ones. There's, I'm going to cover one rare one first, which is fatal familial insomnia. This is a genetic condition where a specific protein creates a compound that builds up in the thalamus, and it's a genetic condition, so it's completely determined by genes, where sleep is completely inhibited, and because of this, if there's no treatment, the individual dies after several months of not sleeping at all. Sleep disorders that affect NREM cycles include insomnia and narcolepsy. Insomnia is a prolonged inability to sleep. There are many different types of insomnia. This can be affected by psychological disorders such as anxiety, depression, and mania. It can also be affected by lifestyle, drinking too much caffeine. Kind of a dysregulated circadian rhythm can also cause insomnia. Narcolepsy is when one uncontrollably falls asleep in the middle of what would typically be wakeful periods. So it's typically 5 to 10 minutes of unexpected periods of sleep. One possible cause of narcolepsy is sleep apnea, which is disruptions in breathing while sleeping, which can cause very poor sleep quality. So sleep apnea can be very, very scary. It can be caused either by the central nervous system or by problems with the sinuses and upper nasal cavity. But people can breathe as little as 4 times in 6 minutes, which you can imagine kind of creates a lot of stress on the brain not having oxygen during those times. REM sleep disorders can be even more bizarre. So one particularly scary one is called sleep paralysis. And this is atonia, which again is complete lack of muscle movement or muscle control, and dreaming while a person is awake. So a person cannot move, they're awake, but they're also dreaming. So they're aware of things moving around in the room. If somebody comes in the room, they'll perceive that to some degree, but they might also perceive that there are demons. it's usually negative things that people with sleep paralysis are perceiving or dreaming about. It has about a 7% prevalence in the general population, so surprisingly large number of people have experienced it. The people that your book mentions that in a student population about 30% experienced it, and I'm very skeptical of that. I wonder if people were misinterpreting the meaning of sleep paralysis. It didn't sound like a very scientific survey. 35% of people with panic disorder actually experience it. So people with panic disorder is surprisingly somatic. It's can be caused by like an overinterpretation of somatic symptoms, and people can have panic when they sleep. They can wake up in a panic disorder, and so associated with this is sleep paralysis. Cataplexy is atonia, or inability to control muscles associated with REM sleep that occurs when a person is fully awake and active. So it's a little bit different than sleep paralysis, but they can co-occur. So cataplexy can be sudden or gradual. Somebody can gradually slump down or just sort of collapse. And this is usually triggered by an extreme emotion. So laughing, crying, fear, embarrassment, surprise. And it's rather common in people with narcolepsy. A lot of these sleep disorders occur together. So if one person has one sleep disorder, it's much more likely that they're going to have another one. Finally, there's REM, or REM sleep behavior disorder, also called REM without atonia. And this is, without atonia means that the muscles are active. And in this, people during REM sleep act out their dreams. So behaviorally enact their dreams. If they're throwing a baseball in their dream, they might actually make that muscle movement while they're sleeping. Sounds like it could be kind of dangerous to anyone sharing the bed with them. Treatment of this includes blocking REM sleep with benzodiazepines, which you can imagine could significantly decrease the quality of sleep that the person is getting, and might have some effects on their life during waking hours. Okay, and then just a reminder, section 7 you don't have to read. By all means read it if you would like, but I won't be covering it in homework or the exam. Okay, so chapter 14, learning and memory, and this is going to exclude section 14.5. I'm excluding it because I felt that we'd already covered a lot of the information included in there, and I wanted to keep this last section brief. So section 1, connecting learning and memory. We're going to be talking about neuroplasticity a lot, which, if you remember from earlier sections, is the ability of the brain to change in its anatomy and or its functionality. This is typically in response to experience or injury, and learning is, of course, a form of neuroplasticity. Learning has corresponding changes in the brain. Learning is the acquisition of knowledge or skills from experience that results in relatively permanent changes in behavior. And memory is the ability to recall or recognize previous experience. Behavioral changes result in brain changes, as I just mentioned. For example, things such as psychotherapy. There's indication that people who undergo psychotherapy and improve whatever emotional or cognitive issues they were dealing with, their brain changes along with that. This includes increases in prefrontal activity, increased connections between brain areas, etc. Alright, so types of learning. I'll go over this kind of quickly. I imagine you learned this in Psych 100. It's just kind of a refresh. So there's the classical conditioning, which is also known as Pavlovian conditioning. This was made famous by Ivan Pavlov, who trained dogs to salivate to the sound of the bell, by accompanying that bell with meat powder, so that eventually, even in the absence of meat powder, dogs would salivate when they heard a bell. So classical conditioning is repeated pairings of an unconditioned stimulus, such as food, like that meat powder, with a conditioned stimulus, which is the bell, and that results in a conditioned response to the conditioned stimulus. So when the bell rings, the dog salivates in the absence of the unconditioned stimulus. Fear conditioning is a form of classical conditioning, and this is when a person or an animal is conditioned to fear a stimulus that would otherwise not be fear-inducing. So there's a famous example that was unethical by all standards these days, but you could do it back in maybe it was the 70s, where a little baby named Little Albert was trained to fear a white rat by associating that rat with a loud sound. So they would show him the white rat and then hit like cymbals behind him, and that would scare him. And eventually they did this repeatedly, and he came to associate the rat with the loud noise, and eventually in the absence of the loud noise, he would still be afraid of white rats. Kind of as a side note, he would generalize that fear. He was also afraid of bunnies and afraid of dogs due to this experiment. And fear conditioning is mediated by the amygdala more than other types of conditioning. The amygdala is highly associated with fear and anxiety and extreme emotions. There's also eye blink conditioning, which is one way that we use to study, it's a more ethical way to study classical conditioning. And this is when a person is conditioned to blink when they hear a tone, because the tone is paired with a puff of air to the eye, so they come to expect the puff of air with the tone, and they blink later just when they hear the tone. And this is actually mediated by the cerebellum because it's motor learning. Blinking the eye is a motor function, and it's one that is mediated by the cerebellum. And then there's operant conditioning, which is another major form of conditioning that you probably also heard of in Psych 100. This comes from learned associations between actions and consequences, so it's more conscious than classical conditioning. I'm not going to go over all of the differences between classical conditioning and operant conditioning, but you can follow this link to a nice little YouTube video if you like. Operant conditioning is it's more complex, and therefore it's less localized in the brain compared to classical conditioning. The areas involved depend on the specific task that's being conditioned. However, the reward circuit is involved, especially in reinforcement. So there's, if you look at this graph to the right, there's reinforcement is what you do to increase the likelihood of a behavior being repeated. So negative reinforcement, you take something averse away, and positive reinforcement, you introduce something that they like. So, for example, if you gave a treat to a dog for shaking your hand, then that would be positive reinforcement. If you if the dog really hates wearing his jacket and you took it off when he shook your hand, that would be negative reinforcement. Something's taken away. And then punishment. Negative punishment is when you take away something that they want, and positive punishment is when you introduce a stimulus that they don't like in order to decrease the likelihood of a behavior. Two main types of long-term human memory are implicit and explicit memory. Implicit is also known as non-declarative memory. Sometimes it's unconscious memory. You can't explicitly retrieve that information. It includes things like riding a bike, conditioned responses, and priming. It conduces procedural memory is a specific type of implicit memory, which is knowing how to do a task without having conscious awareness. So the classic example is riding a bike. If you know how to ride a bike, you can't actually describe to somebody how you ride a bike, like what you do to ride a bike, but you know how to do it. And it also includes priming, which is using one stimulus to sensitize the nervous system to a later presentation of the same or similar stimuli. That's kind of a complex definition for just saying it's kind of prepping your brain for a given response. So there's something called semantic priming, where you prime someone to come up with a certain word. In a semantic priming task, like the one to the right here underneath the bike rider, if you give someone a list of words to remember, you can prime them to help them remember words. So for example, if all these words, garage, nail, violin, and diesel, were in the word list, the word car would prime someone to remember the word garage. Anvil would less strongly but somewhat prime someone to remember nail. However, unrelated words like buckler would not prime someone to remember violin, and cross would not prime someone to remember the word diesel. Okay, these are non-words, so there are non-words included in here. An example of such a lesion study is patient HM, who since his death a few years ago we know is named Henry Molaison, and he had profound deficits in explicit memory. So he had surgery to remove his medial temporal lobe due to seizures, terrible seizures that were affecting his ability to function. So this included portions of the hippocampi, the parohippocampal cortices, endorhinal cortices, and both amygdala. And he suffered from difficulty, or actually complete deficits in forming explicit memory. So other cognitive abilities were retained. He had some implicit memory. He could still remember how to do many skills. And he had memories from before his surgery, but he wasn't able to form new memories. He was profoundly unable to form memories. He would repeatedly ask the name of somebody that he had been meeting every day for weeks. He, if given lunch, would eat lunch over and over again. And he was kind of the first indication that explicit memory relies on the mediotemporal lobe, and that there's this distinction between explicit and implicit memory. So he was probably the most well-known case study within cognitive neuroscience, and was a lifelong research subject who really contributed a lot to the field. Patient JK had a deficit in implicit memory. This individual had Parkinsonian symptoms, which resulted, Parkinson's disease, which resulted in damage to the basal ganglia. It was, he showed deficits in implicit memory, but retained explicit memories. So this indicated that implicit memory relies on the basal ganglia. And again, more evidence that these two systems, implicit and explicit memory, are different. So the brain regions associated with explicit memory, the more specific brain regions, medial temporal regions, as well as the frontal cortices and related areas. This includes the hippocampus, amygdala, parahippocampal cortex, pararhinal cortex, and the endorhinal cortex. That all have specific memories of their own, specific functions of their own. The endorhinal cortex has kind of received more attention lately. It appears to have integrative memory functions, so integrating lots of different memories, and may be the first place that cells die in Alzheimer's disease. So it may be one place that we can target in the treatment of Alzheimer's disease. The hippocampus is highly implicated in spatial memory. So visual spatial memory is the use of visual information to encode and recall the location of an object in space. In London taxi drivers, there's this great study where they looked at London taxi drivers and found larger posterior regions of the hippocampus in London taxi drivers who have extremely good visual spatial memory. London's a huge city, obviously. Within the hippocampus, there are specialized spatial cells that have different functions. And it's a little bit complex, but the three main types of cells are place cells, which are active when an animal is in a given place in the world, regardless of their orientation. So this little rat guy up at the top, the different colors indicate different place cells that fire when the rat is in different areas of this maze that she has run through a number of times. And so this first area that she's in, there are specific place cells that fire when she's at the beginning, these cells that are denoted by this purple color. And then for this other specific place in the maze, there's a different group of place cells that fire for that place. There are also head direction cells that fire when an animal's head is in a specific direction. So regardless of where they are in space, if their head is pointed to the right, then a specific group of cells are firing. And then finally, grid cells fire according to an internal grid. So again, regardless of where you are, of what environment you're in, your brain kind of creates a grid of that environment, and cells fire according to where you are in that grid that you've created in your head. There are reciprocal connections for explicit memory between the neocortex and the entorhinal cortex. The frontal lobe is highly associated with short-term memory. So in addition to the medial temporal areas, sensory areas also, these all project to the frontal lobe. As you know, frontal lobe it has more of like a controlling or executive functioning. So frontal areas are associated with updating and maintaining memories, selecting, manipulating, and monitoring the information from memory, selection of sub-goals that are related to memory usage, so how to use memory to implement goals. The prefrontal cortex is associated with working memory, which is temporarily held information that's used to guide behavior such as decision making and planning. So working memory typically is information that's just being used very briefly. Consolidation is a term for memory that means encoding a memory for storage, or as your book describes it, stabilizing a memory trace after learning. This essentially just means encoding a memory. So when we say if you say, I'm going to try to remember this word, what you mean is you're trying to consolidate that word into your explicit memory. We don't know a ton about consolidation of memories. More research is needed. But there's one theory called the distributed reinstatement theory, that information is learned and then stored kind of temporarily, mostly in the hippocampus. Then this information is rehearsed to strengthen connections between the hippocampus and other areas of the brain, and more long-term memory is stored in non-hippocampal areas of the brain. If you remember the sleep lecture and what we learned about sleep, it's possible that sleep has a role in consolidating the memories and moving it from the hippocampus to non-hippocampal areas. When memories are brought back into our conscious awareness, they connect with new information, they're changed, and then they're reconsolidated. So every time you remember something, you're adding connections to current situations, and you might also be, based on your context or based on other things that you're thinking about, you're changing that memory. And so this kind of speaks to the instability of memory and untrustworthiness of memory. In general, we all tend to trust our memory more than we should. Human memory is unfortunately rather fallible. So for example, in legal cases, when they use eyewitnesses, eyewitness testimony, there are a lot of psychologists who believe that eyewitness testimony should not be as weighty as it is in a legal system, based on information we know about how eyewitness testimony can be rather easily manipulated, even if an individual doesn't mean to. They can kind of convince themselves that they remember something that they didn't. Autobiographical memories are memories about our own lives, and these have a unique neural signature as well. The frontal lobes are involved, as well as the temporal and maybe parietal lobes. There was an individual, patient Casey, who had frontal lobe injuries and couldn't remember any personal memories of life events. So he remembered facts about himself, facts about the school that he attended. He remembered the information he had learned in high school, but he couldn't remember any events that involved him occurring at his high school. There are individuals who have highly superior autobiographical memory, who can recall almost every event in their lives beginning in mid-childhood, and this is associated with increased gray matter in temporal and parietal regions and increased white matter projections between frontal and temporal regions. Perfect memory sounds great, but can actually be a burden to some people who experience it. It can part of the reason that we forget things is because some information is not needed in our lives, and so remembering too much can be certainly a huge benefit in certain situations, but can also have a negative impact on people. Neural circuits for implicit memories include the basal ganglia, which takes input from the neocortex. This then projects to the thalamus and the premotor cortex, and it's associated with dopamine projections through the basal ganglia. And these neural substrates of implicit memories explain why individuals with basal ganglia damage, such as individuals with Parkinson's, have impairments in implicit memory. Emotional memories have a distinct neural circuits as well. Emotional memories don't really fit into the implicit-explicit dichotomy. They could be one or the other. They could be both. It's not entirely clear. They at least tend to have some features of their own. So the amygdala is highly involved in emotion memory, of course. It's connected to the basal ganglia and the mediotemporal cortex. So it's connected to areas involved in both implicit and explicit memories. The amygdala is also connected to the hypothalamus and brainstem structures and the neocortex. So kind of a lot of the major regions of the brain are connected to the amygdala and also involved in emotional memory. Moving on to section 4, the structural basis of brain plasticity. I'm going to go over the section pretty briefly and in broad strokes. I don't want you to worry too much about details in this section unless it's interesting to you personally, because I know it's pretty dense and there's a lot of information in this section, in this lecture already. So neural circuits allow for consolidation and storage of memories, and learning is mediated by changes in these neural synapses within these circuits. Long-term potentiation is a term that we use to describe long-lasting increases in synaptic effectiveness after the synapses have been used a lot, after high frequency of stimulation. So for example, if a synapse is often used, then it's strengthened. There are more receptors on the postsynaptic left. There's more receptors on the postsynaptic cell. Long-term depression is long-lasting decrease in synaptic effectiveness after low frequency of stimulation. So if a synapse is not used often, it's weakened. We sum up that principle by saying use it or lose it. So if a synapse is not often used, if a brain function is not often used, then it's weakened to make room for other synapses. Changes in synapses are reflected in changes in dendritic and axonal structure, mostly dendrites. Dendrites are highly plastic and changeable. We used to believe, I want to say like 30 years ago, that adult brains don't create new neurons, and we are increasingly finding out that that's not true. Adult hippocampus cells, as neurons can be created in the adult hippocampus. For example, in that taxi driver study, they followed those taxi drivers for I think like 4 years and found that over the course of training, taxi drivers had increases in hippocampal volume. As well, new neurons are found in frontal regions, temporal, neocortex, and the olfactory bulb. Changes in the brain are, of course, dependent on experience. So an enriched experience leads to increased neural growth and synaptic connections. We saw some of that in the developmental chapter, where kids who grew up in the Romanian orphanages had less brain growth than kids who grew up in more enriched environments where people were paying attention to them, their social interaction, etc. Increased complexity and expertise in a skill is associated with more neurons and connections in that region that's dedicated to that skill. And higher education is associated with more dendritic branching in Wernicke's area. So higher education, presumably you're using your language skills more or developing them in a highly skilled manner. And so we're seeing language areas of the brain having more dendritic branching, more associations in those areas. Just a note, I'm skipping material from the book for the rest of the section, so I'm skipping around in the section a lot to kind of try to keep the information that you have to deal with to a minimum, because I know there's a lot of information in these two chapters. So the general principles of brain plasticity: behavioral changes reflect brain changes. All nervous systems are plastic in the same general way, so plasticity in the human brain looks like plasticity in the rat brain or the monkey brain. Plastic changes depend on age. Young brains are very plastic. Older brains are definitely less plastic. Prenatal events can affect later potential for plasticity, so adverse prenatal events, as well as exposure to neurotoxins, can affect the ability of the brain to be plastic later on. Plasticity differs among brain regions, so we talked about how the hippocampus has a high potential for plasticity, whereas other areas of the brain are less likely to change. Experience-dependent changes interact with one another, so different areas of the brain, depending on what's in one area of the brain, changes may be different depending on what's happening in another area of the brain. And finally, plasticity can be a benefit or a hindrance to humans, and we can learn bad behaviors, and it can be hard to change those learned bad behaviors. So this is chapter 15, Thinking in the Brain, and I've excluded I've excluded 3 of the sections, 4, 5, and 6, for the sake of brevity. I think a lot of the materials that were in those sections we did touch on in previous sections of the book, but by all means read those sections if you are interested. So the nature of thoughts. This chapter, I think, ended up being a bit of a catch-all, just covering a lot of topics that didn't necessarily fit in other sections. So it's going to jump around a little bit, but there's definitely some interesting material. Psychological constructs. A construct in psychology is idea that a mental ability is an actual entity. So the idea that like memory is actually a thing, or language or emotion. Before we knew about brain systems, we had all these kind of folk psychology terms for things, and the more we move into brain systems, some of those constructs in psychology are being reinforced by brain areas that are dedicated to those brain areas and systems that are dedicated to those functions, whereas other psychological constructs we're not finding to be explicitly represented in the brain as well. We don't always have consistent definitions of psychology constructs, which makes psychobiology really complicated, because if we can't define the construct, it's hard to say where it's represented in the brain. So for example, thought is a difficult construct to pin down. In fact, many people wouldn't use that term in psychobiology because it's so vague. A thought can mean something that you're aware of. A thought can be unconscious or conscious, depending on how you define it. It can cover a lot. It can be a very specific thing as well. Similarly, like intelligence, there's a lot of controversy over how intelligence should actually be defined, and a range of other constructs in psychology as well. There are some characteristics of human thought that we have observed. One is that it appears to be mostly verbal. This is very interesting. So one suggestion for why babies and very young children don't form explicit memories is that they don't have language yet, and the way that we actually think in language even if we're not fully aware of it. Language allows us to organize time and ourselves. Kids self-talk a lot when they're young, so you might see a little kid when they're playing. If she's working with her blocks, she might talk to herself and say, stack this block on top of this block. Kids talk to themselves a lot more, which eventually turns into subverting self-talk. So as we get older, we stop talking to ourselves. But a lot of that self-talk is still happening in our thoughts. Borrowing from the field of schizophrenia research, this is actually likely where command hallucinations, which is hallucinations where a voice is telling somebody to do something. This is actually just a misinterpretation of self-talk. Actually, we kind of verbally command ourselves to do things a lot, but we're not aware of it. We're not consciously aware of a lot of our self-talk and how verbal a lot of our thought processes are. Syntax, which we talked about how syntax is innate and it supports this ability to organize our thoughts. The caveat is that we have to learn language in order for our innate syntax to emerge and help us organize our thoughts. So if someone did not learn language by the critical period by around 6 years old, they won't have that innate syntax emerging, and they won't be able to organize their thoughts based on language. Another characteristic of human thought is that the human brain wants to make meaning out of its surroundings. It's constantly trying to make meaning. One kind of consequence of this may be this déjà vu, which is this feeling that you've done or heard something before, but you can't quite place it. And this actually might be an example of kind of spurious attempts of the brain to create connections. Our brain is always trying to create connections. So you might occasionally have this feeling of having experienced an exact moment previously. And thinking is likely interaction, however we define it. It's probably interactions between many different brain systems. There's no one thinking region of the brain. Association cortices are something we didn't get to talk a lot about. They were mentioned kind of briefly in several chapters, especially the sensory chapters. But these are neocortices outside of the primary sensory and motor areas. So there's the motor association cortex and the sensory association cortex, visual association, etc. Input to the association cortices comes from parts of the thalamus that also receive information from other cortical areas. So association cortices may be integrating sensory information with higher level information, other cortical areas. In addition, we didn't get to talk much about the anterior cingulate cortex, which is this pretty interesting medial part of the cortex. Some people consider it part of the frontal lobe, some people don't. But it has a connection, creates connections between emotional systems and cognition, and has a lot of interesting functions that we don't have time to get into right now. But it does seem to be another area that kind of creates a connection between other functions. Multisensory integration is another interesting area that we didn't get to talk about. So our brain integrates information from different areas really quickly and effectively, to the extent that we don't really know how to explain it, and this is called the binding problem, explaining how our brain is able to integrate information so quickly and effectively. We understand a lot of the more basic processing better, but we don't understand the really higher order processing. Possibly multimodal regions of association cortices could be involved in multisensory integration and the binding problem. Multimodal regions integrate information from more than one sensory modality, so integrating information from visual and auditory systems, for example, We don't have a good map of this in the brain, but there's this image from a monkey brain of some areas where it's been noted that more than one sensory modality is being processed in that area, at least sometimes. Many of you might have heard of synesthesia. Some people are synesthesiacs, which means that when one sensory modality is stimulated, it automatically stimulates another. So I think one of the more common ones is grapheme color synesthesia, in which an individual, if they see a letter, specific letters and specific numbers each have their own color. So S's in this example are always this fuchsia color, A's are always this dark red. Numbers are like the 1 is always a teal color, for example, and it's kind of like automatically processed this way. There's indication that this may result because of cross-activation of the color and grapheme areas in the brain. They're pretty close to each other, grapheme being symbols like numbers and letters. They're close together, and so in some people's brain, these may get activated at the same time. There's also there's a number of types of synesthesia, and it's really interesting. I recommend reading more about it or checking out YouTube videos about people who have it if you're interested. But there's another type called mirror touch synesthesia, where a person feels sensation that they observe in another person. And this is might be related to empathy, but it more typically occurs in people who have other types of synesthesia as well. Attention is a cognitive function we also did not get to talk about. Attention is when the focus of awareness is the ability to focus awareness to internal or external stimuli. So you can focus your attention inward to try to get a memory, to try to think about something, to try to plan or decide. You can also focus attention externally to something in your environment if you want to focus on your computer screen, for example. It can be influenced by factors in the environment as well as internal influences. So in our environment, there are certain things that naturally catch our attention. If there's movement in our environment, in an otherwise still environment, this automatically catches our attention. Novel stimuli, things we don't recognize. If we're sitting in a classroom and been sitting in the classroom with the same people every week, and there's a new person suddenly, this person is more likely to attract our attention, kind of naturally and almost against our will. Internal motivation is another way of internally allocating attention. So if we have an internal motivation, if there's a book we really, really want to read, this is kind of like an intrinsic motivation to focus our attention on the book. To the right, these dot pictures, this is just an example of how certain novel stimuli are likely, or novel meaning like new or different stimuli, are more likely to attract our attention. So in the image where it's all green and there's one red dot, you can't help but look at the dot. It's almost impossible to ignore that dot. Whereas in the right panel where there's many different colors, you actually kind of have to try to find the red dot because there's many novel stimuli, or it's not that different. And this is called the pop-out effect. When there's one thing that's quite different, it grabs our attention. Attention has pretty complex neural underpinnings. The dorsolateral prefrontal cortex, as well as frontal eye fields, which are just a little bit anterior to the DL-PFC, are implicated in attention, but a number of other brain areas are as well. Ultimately, stimuli are fighting for our attention, so stimuli in the environment and also internal stimuli are constantly engaged in a competition to be present in our conscious awareness. Okay, so moving on to section 3, expanding the frontiers of cognitive neuroscience. I picked out, I didn't include most of the information from this section, but I thought social neuroscience is very interesting. And this is the study, of course, of social functions and social phenomena in the brain. Theory of mind is the theory I mentioned in a previous lecture. So it's the ability to attribute mental states to other people. people with autism tend to be poor at this. They're not able to guess what another person is thinking, and difficult to understand complex social interactions. And there are specific neural areas that are associated in theory of mind. One is the dorsomedial prefrontal cortex. Another is the temporal lobe, temporal pole, and then another part of the temporal sulcus. The sense of self, or understanding of oneself, is also, of course, represented in the brain. There are many areas of the brain that code for different types of sense of self, because there's actually many components of having a sense of self. There's a physical sense of self, sense of owning your body, sense of controlling your own actions. It's actually a really complex construct. But some of the main areas in the brain that have been associated are represented by these colored rectangles in the midline structures of this brain. So some frontal regions and then parietal regions as well. And then finally, there's this one area of the brain in this represented in this spinning brain. The little red dot is out-of-body experiences are in the temporal parietal junction. So this is the connection between feeling like you're embodied within your body. So out-of-body experience is something people sometimes have when they're on the verge of dying, or it can actually be stimulated. It can be stimulated by TMSing the temporoparietal junction, but it feels, some people have felt that they're in an out-of-body experience. It feels like your soul, for lack of a better word, is exiting your body. It feels like you no longer inhabit your body. And that is a rather specific area of the brain. Okay, so the last section of the class is consciousness, and this is really complex. The neural substrates of consciousness and the neural underpinnings of consciousness are, it's one of the most complex areas of neuroscience in general, and but part of this is because consciousness itself is a very complex construct. It's been defined in many different ways. Your book defines it as the mind's level of responsiveness to impressions made by the senses, and I think this is a very neuroscience-based definition of consciousness. Other people define it as the ability to think derived from the ability to feel, or the awareness of one's own existence. Some people say it's whatever, take animal cognition and some practice from human cognition, and whatever's left over is consciousness. Notably, many people in psychology don't like the term consciousness and don't believe that it's a meaningful term or a meaningful construct. People have told me that it's a 4-letter word in psychology. But there are scientists who have attempted to study consciousness in the brain and have come up with some pretty interesting results. So this individual, Christoph Koch, he's one of the most well-known researchers of consciousness, and he's a really unusual guy with an affinity for pink. He had his hair dyed pink for some reason for a really long time, and he forces his lab to wear these unusual red vests because he's obsessed with color, but he's all over just a really unusual guy. But I recommend, if you're interested in the neurobiology of consciousness, I recommend looking up. I'm sure he has TED Talks. But Christoph Koch has studied the neurobiology of consciousness throughout his entire career, and he believes that there's this area called the claustrum, which is the most likely candidate for as a neural substrate of unified experience and the regulation of consciousness and cognition. He believes that it might use synchronized neural oscillatory activity to unify brain areas. So neural oscillatory activity is when lots of neurons fire together, and this can happen across a range of brain regions, and they fire together just like in sleep waves. We talked about oscillatory activity, the alpha waves and the beta waves, things like that. So fire together, and this may be a way of sort of unifying areas, activating areas of the brain simultaneously. There's also, as I mentioned in the optional readings for this section. There's a recent article by Christoph Koch's group where they recently found neurons in the mouse brain that are encircling the entire brain. So it's really, for the central nervous system, a really long neuron. So for interneurons, it's a really long neuron, and appears to have connections within quite a lot of different areas of the brain. And Christoph Koch's group thinks this may be somehow related to consciousness and with unifying different areas and functions of the brain.